Good evening, everyone. Welcome to week 9 of Living Systems. Tonight we'll be looking at lecture 12, Movement Across Membranes. We're going to focus on biological membranes in this very important lecture. And we can see on this on this first slide here, a biological membrane. So this might well be, for example, the lipid bilayer that you would find in the plasma membrane. And what we can see here is an example of a membrane protein. This particular protein here is a transmembrane protein because it extends right through the membrane from one side to the other. You'll see also that there's a channel which allows the direct movement of molecules from one side of the membrane to the other. So we're going to talk about this type of transport process that exists, that cells use, and also other mechanisms which cells use to transport substances from one side of the membrane to the other. So let's start by considering this cell here. An example of a cell, obviously a red blood cell, and we'll look at the concentration of 3 ions. Potassium ion is present at a concentration of about 140 millimolar inside the cell, but outside the cell in the plasma, so the fluid component of the blood, it's found in very low concentrations. But if we look at sodium and chloride ions, they're present at high concentrations in the plasma, but really low concentrations inside the actual cell. So obviously this suggests that there must be one or more mechanisms that the cell uses to maintain these different concentrations, and that's the focus of tonight's lecture. So plasma membranes control the passage of substances into and out of a cell. And this is important because cells, or rather the reactions which occur within cells, operate within a very narrow range of conditions. So maintenance of stable conditions inside the cell is referred to as homeostasis. But remember, it's not just the plasma membrane itself that's important. There are also membranes surrounding structures inside the cell, that is organelles. And these membranes also function in the same way to control movement of substances. So let's consider now the permeability of different molecules. That is how readily they can just move directly through that lipid bilayer of a membrane. Permeability of molecules depends on their size. So the larger molecules will have a lot of trouble getting through directly through the lipid bilayer of a membrane. Smaller molecules have a greater chance of passing through. Molecules that are charged, any charged molecules such as ions, as we can see here, will also have problems getting through the lipid bilayer of a membrane. And that's because you might remember that the interior of that lipid bilayer is highly hydrophobic. And this would pose a resistance to any charged molecules that are trying to get through. In contrast, molecules that are soluble in lipids would have a better chance of interacting with that lipid interior of the lipid bilayer of the membrane. So they would have a greater chance of moving through the biological membrane. Okay, so let's look at this term diffusion and consider the diffusion of solutes in water. So how solutes move from one solution, for example, through a membrane, through a biological membrane to the other side. So diffusion can simply be defined as the passive movement of molecules along or down a concentration gradient from high concentration, from a region where that molecule is found in high concentration, to a region where that type of molecule is found in a low concentration. So the best way to actually visualize this is to consider a glass of water, and that's supposed to be wavy. My drawing is just fantastic. Glass of water, and let's say we place into that water a drop of dye, drop of food dye. So obviously where we place that drop of food dye, that's where the dye molecules are found in high concentration. Now, what happens to those dye molecules? They will start to spread out. Why will they spread out?
Oh, because it's diffusion. So you get high concentration moves to lower.
Correct. Yes, but what's making them move? So they are moving from a region of high concentration to regions of low concentration. They'll simply move out passively and randomly without any input of energy whatsoever. And what makes them move? Something that we learned about in a previous lecture. All molecules have an intrinsic ability to move, don't they? Kinetic energy of motion. Yeah. So they'll simply move around at random, and they'll continue to do so until an equilibrium is reached, until they're all spread out evenly throughout that water. How would we increase the rate at which they move around, the rate at which they diffuse or move from high concentration to low concentration? What can we do to increase the rate at which they diffuse?
Heat, use of heat.
Heat, yeah, absolutely. So if we increase the temperature, then obviously the movement of those dye molecules is faster. They'll diffuse much faster through the water. Now, what would happen if we drop the temperature right down to 0 Kelvin? That looks very much like okay, doesn't it? It's supposed to be 0 Kelvin. What if we drop the temperature down to 0 Kelvin, or what's the equivalent in degrees Celsius?
Minus 273.
Minus 273 degrees Celsius, and then we added that drop of food dye. What would happen, do you think?
Nothing, isn't. That's where motion ceases.
Correct. So if that's the temperature, the theoretical temperature at which all molecular motion ceases, then you would expect that that drop of food dye would just simply remain intact. The dye molecules would not diffuse because they no longer have any kinetic energy of motion. All right, so that's diffusion, passive movement of molecules. And of course, when we say passive, we mean that there's no input of energy required for that process to occur. All right, so diffusion of certain substances, passive diffusion of certain substances does occur across biological membranes. Examples here, oxygen, carbon dioxide. This is how they always move into cells or out of cells. That's how these molecules move. There are no special transport proteins embedded in membranes to move oxygen and carbon dioxide. Alcohol, so ethanol and some of the other smaller alcohol molecules also readily diffuse directly through the lipid bilayer of a membrane. The rate of diffusion depends on, well, mostly on two factors. One is the permeability of the actual substance that's diffusing. The higher the permeability, the faster it will move across. And also the rate of diffusion depends on the magnitude of the concentration gradient. So if you have a really high concentration on one side of the membrane and a really low concentration on the other side of the membrane, then you've got this, this high magnitude, this great magnitude, and that increases the rate of diffusion. All right, let's clear that. And here I'm going to put I'm going to draw very roughly a lipid bilayer, because this is our number one transport mechanism directly across the membrane. Okay, so number one is passive diffusion from a region of high concentration directly through one side to the other without the aid of any transport proteins to low concentration. Okay, there we go. All right, diffusion of ions occurs along an electrochemical gradient. So here we don't just consider the concentration, but also the electrical difference across the membrane. So the electrochemical gradient is a combination of the concentration gradient, or the chemical gradient from high to low concentration, and it also includes the electrical gradient, which is the difference in charge across the membrane. And this difference in charge always exists in cells, across the plasma membrane, and also across the membranes of organelles. So remember, when we're talking about differences in charges across the membrane, opposites will attract, as in species with opposite charges will attract, and those with like charges will repel each other. The direction of net passive movement across the membrane will depend on the relative strength of these two gradients. So let's illustrate that on the next slide. So in this upper panel here, we're considering only the chemical or concentration gradient on its own, and we have here a cell. And outside the cell, we have a high concentration of molecules, low concentration here. So, of course, because diffusion occurs from a region of high concentration to a region of low concentration, these molecules will simply diffuse into the cell, which is what you can see here, until there's an equilibrium reached. But of course, cells avoid these equilibrium states at all times. They're always pushing every process out of equilibrium, because you know that's when work occurs, when processes, reactions are not at equilibrium. All right, in the bottom panel now, we can consider an electrochemical gradient instead of just a concentration gradient. Once again, we have a cell pictured here, and now you can see that just to the inside of the plasma membrane, there is a negative charge. And this is quite the standard for cells, to have very slight negative charge just inside the plasma membrane. It doesn't mean that charge extends right through the cell. Charges can be very localized to certain regions of a cell. Now, let's consider these molecules to the outside. They're obviously ions, in red, positively charged, and in blue or purple, negatively charged ions. So what do you think will happen given that we have a high concentration of ions out here, but we have negative charges just to the inside of the membrane? What do you think will happen in terms of diffusion?
The cations will move across because they
correct.
Attracted.
Yes. Yep, absolutely correct. The cations in red are the ones that will most likely move into the cell, because, as we said earlier, opposite charges attract, like charges will repel each other. And that's what you can see here. Predominantly positively charged ions have moved into the cell to balance out those negative charges. So this is an example of both electrical and chemical equilibrium. But once again, this is not a desired state for the cell. All right, now let's look at the second mechanism that cells employ. So mechanism number two And I'm going to draw my membrane in a very messy fashion. Here's my lipid bilayer here of the membrane. And once again, we're going to look at movement down a concentration gradient from high to low. But this time, this process involves the use of transmembrane proteins. So it's actually referred to as facilitated passive diffusion, because that diffusion across the membrane actually employs the use of membrane transport proteins, membrane transporters. Membrane transporters will accelerate the movement of less permeable molecules across the membranes. These transport proteins are specific, highly specific to either one molecule, one type of molecule, or a very small number of molecules. Let's look at an example of how efficient this system is of facilitated diffusion or facilitated transport. So if we look at this graph here, on this axis we have the rate of glucose uptake into a cell, and on the x-axis we have an increasing concentration of glucose. So this is the external concentration of glucose using the units millimolar. Now let's consider just simple diffusion directly through the membrane, which is what we have up here. So without the aid of membrane transport proteins. And you can see that there's a very, very low rate of glucose uptake, even at really high concentrations of glucose. But when glucose moves through a membrane transport protein, which there are many different membrane transporters that are specific for glucose, then look at the rate of uptake. Even at very low concentrations of glucose, it is very, very efficient until it gets up to about here, and then it starts to level off. And the reason it levels off is because the rate of transport across the membrane depends on the number of transport proteins for that particular molecule that exists in the plasma membrane itself. So imagine if all the transport proteins that are specific to glucose are all occupied, they're all transporting glucose as fast as they can go, then we'll reach that saturation point and the rate levels off, as you can see here. Okay, so that's our second mechanism. It's called facilitated passive diffusion. So it's still passive, still doesn't require an input of energy by the cell, and it still follows or requires that concentration gradient. But it's just much faster and more efficient because the substance is moving through a specific transport protein. All right, so still on facilitated diffusion, transport proteins assist the movement of molecules down a concentration gradient through facilitated diffusion, requiring no energy input. There are two broad categories of membrane transport proteins: channels and carriers. Channels allow the direct passage from one side of the membrane to the other, whereas carriers are a little bit different. They will bind the molecule on one side of the membrane. This binding produces a change in shape of the membrane transport protein, otherwise known as a conformational change, and this allows the actual molecule to move to the other side of the membrane. And we'll look at images of these in just a moment. So the first broad category of membrane transporters are the channels. These are quite rapid. They allow molecules to move in and out of cells quite quickly. They're faster than carriers, which have to bind and then release solutes. Most channels that have been studied are ion channels. They transport different ions. They have high specificity, usually for one or two particular types of ions. And here are just some of the ion channels that have been studied. Calcium channels, potassium channels, chloride channels. But there's quite a few others. All right, so this is what a channel protein looks like. And we saw this on the very first slide. So here it is, a transmembrane protein, which is a channel protein. This is the closed state. And here we can see the open state. So when the channel's open, whatever substance it's specific for can simply diffuse straight through this open channel from one side of the membrane to the other. But remember, diffusion occurs still down a concentration gradient from high concentration to low concentration. Now let's compare that to a carrier protein. So here's a carrier protein which is open. So here's the pocket. It's open just to one side of the membrane in this state. The solute can bind. There it is, bound in that pocket. The binding of the solute or solute molecules causes a conformational change in the transport protein. So here you can see it's changing shape. Here it is again, changing shape. And this time that pocket is now open to the other side of the membrane and it can release the solute to the inside or to the outside. So that's a carrier protein. All right, let's go back to those channels that simply have the open and closed states. Channels are opened and closed by various signals, depending on what type of channel it is. Here are the three major types of channels relating to what those signals are. We have voltage-gated channels, which respond to changes in voltage across the membranes. We have ligand-gated channels, which are activated by binding of specific molecules called ligands. And we have mechanically gated channels, which respond to physical disturbance. So if you're interested, you can look those up and you'll be able to read about lots of different types of signalling processes that cause these channels to open and close. All right, so let's wrap up facilitated passive diffusion. Properties of facilitated transport that distinguish it from simple diffusion. Transport is faster. Transport proteins will become saturated as the substrate concentration increases. Transport proteins have high specificity for particular substrates, and transport can be inhibited by similar substrates that compete for the binding site. So if we think about certain types of poisons, this is how they act. They structurally mimic other molecules and then compete for binding sites, which means that transport protein can't do its job. All right, now we move on to our third transport mechanism. So number 3. And here's my lipid bilayer, and my drawing's getting worse all the time. There's my membrane. And this time we're going to look at a process. Oops. We're going to look at a mechanism here, high to low. So instead of molecules or substances moving from a high concentration to a low concentration, this is a mechanism that enables the movement against the concentration gradient. So from a low concentration to a high concentration. So active transport means that energy is required for this transport process to occur. Active transport uses energy to move the solute against its concentration gradient or against its electrochemical gradient. In other words, the energy that's normally used is our adenosine triphosphate or ATP. So one category of transport proteins that are involved in active transport are ATPases, transport ATPases. Here's an example down here. This membrane transporter is an ATPase, and it is specific for the transport of calcium ions. It requires energy from ATP, so ATP has to be hydrolyzed to ADP in order to push or directly pump calcium against its concentration gradient across the membrane. So that's one type. Another type of active transport mechanism that's commonly used is called co-transport. This involves the diffusion, so just the passive diffusion of one molecule down its electrochemical gradient, and that passive diffusion is used to actively pump a second molecule against its electrochemical gradient. So let's look at this process here on the right, and this system that we're looking at is very commonly employed in plants. And we'll look at the system, another system that's used in animals in just a minute. All right, so let's start here with another type of ATPase, and this is a proton ATPase. So obviously this is a membrane transporter that actively pumps protons against the proton concentration gradient. In other words, from low to high. It does this, of course, using energy provided by ATP. So the buildup of protons on one side of the membrane means that given the opportunity, those protons will do what? They will just naturally diffuse back down their electrochemical gradient. But they can't do that directly through the membrane itself. Protons are not very good at diffusing through membranes, but they can diffuse, passively diffuse back down their electrochemical gradient through another membrane transporter here, which is called a symporter or a symport transporter, symport protein. Okay, so this type of symporter allows the protons just to passively diffuse down their electrochemical gradient. And here's the interesting bit, the movement of protons through here provides the energy to move another molecule, in this case amino acids, from one side of the membrane to the other using the same membrane transporter. So this is referred to as a symporter because it's allowing two different molecules to move in the same direction across the membrane utilizing the one membrane transport protein. Let's compare that to this one here, which is called an antiporter, an antiport protein. So once again, the protons are simply diffusing back down their electrochemical gradient. The movement of those protons provides the energy to transport a different molecule, in this case sodium, and sodium can move through the antiporter but in the opposite direction. So this is why it's called an antiporter. So that's co-transport in a nutshell. Let's revise that on this slide. So in plant cells, protons are pumped from the cell using the proton ATPase that we just looked at, and this creates an electrochemical gradient. So the protons then move back into the cell, just passively diffuse back into the cell, and this movement of the protons drives pumping of other solutes, such as the amino acids or the sodium ions, against their electrochemical gradients. In animal cells, rather than using these gradients of protons, gradients of sodium are established, and these are established by a different ATPase membrane transporter called a sodium-potassium ATPase. And we can look at that on, not that slide, but the next one. I'll come back to this slide in just a minute. All right, here we can see how the sodium-potassium ATPase works in animal systems. So quite a busy slide, so let's just focus our attention on this membrane transporter. So this is the sodium-potassium ATPase. And you can see that it's open to one side of the membrane, so this opening is facing the inside of the cell. In this particular conformation, it will bind 3 sodium ions, and we can see them here in green. So they're bound to the ATPase. Once they bind this, once they bind, then ATP is hydrolyzed to ADP. The phosphate that's released from the ATP covalently binds here to the ATPase. And that binding of the phosphate triggers a conformational change in the ATPase so that now the opening faces the outside of the cell. This enables the sodium ions to be released to the outside, and once they're released, then potassium ions can be bound. 2 potassium ions for every 3 sodium ions. So they're illustrated here by these little red triangles. Once they bind, then this triggers, again, a conformational change in the ATPase. So we go back to the original conformation here, where the opening faces the inside of the cell, and those 2 potassium ions are released to the inside, and the phosphate is also released. Now, once the ATPase is in this conformation, then it can start the process all over again. So this explains, I think, quite nicely how cells, or at least animal cells, maintain a high intracellular concentration of potassium ions and a low intracellular concentration of sodium ions, while maintaining high external sodium and low external potassium. All right, sorry, I need to go back one slide. I really should change the order of these slides, but I never think of it until I actually deliver the lecture. All right, so as we said earlier, there is a voltage difference across a membrane. So on one side of the membrane compared to the other side. And this difference in voltage is referred to as the membrane potential. So what sets up that membrane potential in the first place? The membrane potential is due to the negative charge of certain proteins and other polymers that are inside the cell, and also the transport of ions across the plasma membrane contributes to maintenance of that slight negative charge, somewhere in the order of about negative 50 millivolts, but that also depends on the compartment within the cell. So removal of positive ions from the cell also increases that negative charge just to the inside of the plasma membrane. All right, why are these membrane potentials important? They provide a favourable electrochemical gradient for passive uptake of cations. They provide an inward electrochemical gradient which can be used in actively transporting other ions, and changes, even very slight changes in the membrane potential, are very important in terms of signalling. Signalling can be local within the cell, or the signals can be transmitted from cell to cell. Clear all that now. All right, so now we're going to look at the important concept of osmosis. So osmosis is almost identical to diffusion, except that now we're just talking about movement of water molecules rather than when we discussed diffusion, we were looking at movement of solutes in water. So water will travel passively across biological membranes, and it does this by the process of osmosis. Water will move from a region of higher free energy to a region of lower free energy. And I'll show you this in just a moment. The free energy levels of water are determined by, one, the concentration of dissolved solutes in that water, and particularly for plant cells, it's determined by physical pressure or tension. The free energy levels of water are decreased by increasing concentration of solutes and decreased by reduced pressure, particularly in plant cells. All right, so let's look at osmosis. To the right here, we have, to the right, of this selectively permeable membrane, we have only water molecules. So let's think of this as pure water. In this example here, where you have only pure water molecules, water is said to have the highest possible level of free energy, and that's given a value. What is that value? The value is 0. It's set at 0 megapascals. 0 megapascals. That is the highest free energy level of water. In other words, those water molecules have the maximum capacity to move, maximum amount of kinetic energy. They're not inhibited by anything or restricted by anything. All right, to the right of this membrane, you can see that the water molecules, or many of the water molecules, are interacting with these solute molecules. So given that they're being bound up via those interactions, so you can think of the solute molecules as sodium ions, chloride ions, or anything that will interact with water molecules. So as I was saying, because these water molecules are interacting with the solutes, they have less free energy. And so we can say, for example, that the free energy of water on that side of the membrane will be lower than the maximum amount of free energy. So it might have a value, the free energy value might be negative 1 megapascals. Okay, lower free energy. So when we talk about osmosis, water will always move from a region of higher free energy of water to a region where there is a lower free energy of water. Okay, and that's summarised here. So water will flow across a membrane towards a region of higher solute concentration, which is the same as saying the lowest water free energy. In plants, as I said earlier, pressure is also important, and we'll illustrate that in just a moment. So, free energy of water, the overall free energy of water is referred to as the water potential. And water potential is given this symbol, which is called psi. And I'll just write that here. There it is, psi. And the water potential is a sum of the osmotic or solute potential and also the pressure potential. So that's illustrated here. Overall free energy of water, or the water potential, is equal to the osmotic or solute potential plus the pressure potential. And I'll just mention here that the symbol for osmotic or solute potential for some of you at least in the future, you'll see it written like this, just with a lowercase s. Okay, particularly for those students who will be doing plant physiology in their second year. It's simply referred to as the solute potential or the concentration of solutes. All right, so now we can define osmosis. Osmosis is the net passive movement of water from a region of higher water potential, remember, high free energy of water, to a region of lower water potential through a selectively permeable membrane. And now that we understand this, we can actually work out what happens when we place a cell in a solution with varying concentrations. So if we place a cell into an isoosmotic solution, also referred to isotonic solution, then that solution will have the same solute concentration as the solute concentration that you'll find inside the cell. If we place a cell into a hyperosmotic solution, that means that the solution is more concentrated than what you find inside the cell, and a hypoosmotic solution will be less concentrated. All right, so let's start with animal cells and those three different types of solutions. And this is all very relevant to a prac that some of you have already done, the cell membranes prac. And some students, weekly prac students, will be doing in two or 3 weeks' time. 2 weeks' time, actually. All right, so what happens when we place an animal cell into a hyperosmotic solution? Okay, so let's take a solution here. And we'll place our red blood cell in here. And the solution has a high concentration of solutes because it's hyperosmotic. High concentration of solutes in this solution compared to what's in the cell. In which direction will water move?
It will leave the cell.
It will leave the cell. Water will move from inside the cell to outside the cell. So it's actually moving from high water potential here, that is high free energy of water here, to lower free energy of water here. Or, of course, you can look at it in another way. You can say that water will move from low solute concentration to high solute concentration here. So what happens to the cell when it loses water? It will shrivel up, as you can see here. All right, let's try that again, and this time with an isoosmotic or isotonic solution. Okay, once again, so this time the solute concentration in the water is the same as the solute concentration inside the cell. So in other words, it's probably something equivalent to about 0.9% sodium chloride, which is something that you would have used in your cell membranes prac as an isotonic solution. So what happens in this case to water movement?
It's an equilibrium. So
yes, so it'll be the same in both directions. Yep, there'll be no net overall movement, and the cell, the animal cell, will retain its normal shape. All right, the last example, hypoosmotic solution. Okay, so this time we're placing the cell in go, there's our cell, and now we're placing it into a solution that has a very low solute concentration. So for example, 0.1%. 1% sodium chloride, which is much lower than what you would find inside the actual cell. So what would be the direction of water movement here in this case?
Going into the cell.
Going into the cell. Water will move from the solution into the cell, and that causes the cells to swell up, and you can see that here, swelling up and eventually burst. Now let's compare that to plant cells, which respond a little bit differently. All right, here we have plant cells. Once again, we start with our hyperosmotic solution. And what happens to our plant cell when we place it in a hyperosmotic solution? A little bit different again, once again, to animal cells, because plant cells are surrounded by this cell wall. So the cell wall remains relatively unchanged, but the plasma membrane will start to shrink away from the cell wall as it loses water. Here we have a plant cell that's been placed in an isoosmotic solution. Okay, so because there's no net change in the water that's moving into the cell or out of the cell is actually flaccid, which is not the desired state for a plant cell. To the right, we have the desired state for a plant cell. A plant cell that's surrounded by a hypoosmotic solution will result in the inward movement of water into the cell, and of course the cell will start to swell just like an animal cell does, but it will not burst because the cell wall also expands by about 10%, and then it resists any further inflow of water. This means that we have pressure building up inside the cell, and that's required to facilitate this normal turgid cell state within a plant. So once again, quite different to animal cells. Okay, let me get rid of this. Okay, so in animal cells, the differences in water potential across the plasma membrane are due only to differences in osmotic potential. That is, differences in solutes on either sides of the plasma membrane. But when we're talking about cells with a rigid wall, whether it's plant cells or cells of algae, cells of bacteria, and so on, then both the osmotic or solute potential and the pressure potential will contribute to the overall water potential. So water will enter when cells are placed in solution with a less negative water potential. That is a solution that is hypoosmotic, but the cell only expands by about 10% before that internal pressure, which is referred to as turgor in plant cells, causes that intracellular water potential to equal that of the external solution. So the shape of plants is maintained by cell turgor, and this is part of the reason why plants constantly have to take up water from the soil solution. That water that's taken up by the plants, some of it's used to maintain cell turgor, but a lot of it actually moves through the plant and then out into the atmosphere. And that's because the atmosphere has a very, very low water potential. If the lost water is not replaced, then the pressure potential inside the cell, that is the turgor pressure inside the cell, is reduced, and of course that leads to plant wilting. And plants that are wilting will shut down those important metabolic processes that enable growth and development to occur. Okay, and finally, we're going to look at another type of transport mechanism, which you've already been introduced to in your functioning cells lecture, and this is vesicle-mediated transport. So larger molecules are transported in membrane-bound vesicles. And you might remember from last week that we described vesicles. So for example, this might be your Golgi apparatus. Oh, and you remember that we said that little bits of membrane from the Golgi will bud off and then pinch off to form these tiny membrane enclosed vesicles. And of course, these vesicles are carrying some type of cargo. All right, two types of vesicle, or two broad types of vesicle-mediated transport. One is called endocytosis. So this is where the plasma membrane of the cell will actually enclose substances that are found outside the cell. And then those little bits of plasma membrane will pinch off to the inside of the cell to form that little vesicle. If the vesicle contains solids, then it's referred to as phagocytosis. If the vesicles contain fluids, it's called pinocytosis, or commonly referred to as cell drinking. The invagination of the plasma membrane may be receptor mediated, and I'll show you that on the next slide. All right, so here we have an illustration of endocytosis. Right, here we have the plasma membrane of the cell, and you can see that inward movement called invagination. And the cell in this case is being tricked to invaginate by the presence of certain bacterial cells. This is how some pathogenic bacteria actually gain entry to cells and infect cells. All right, further invagination occurring there and here. And it's nearly ready to pinch off. Here it has pinched off to form a vesicle, and the bacterial cell has entered. All right, what we're seeing here is an actual image of this process, a real image taken with an electron microscope, but it's just been coloured. All right, another example here of endocytosis, and here we have some solutes that are found in high concentration to the outside of the cell. Once again, invagination, endocytosis to form that vesicle. And here we have an example of receptor-mediated endocytosis. So here we have some proteins embedded in the plasma membrane, but these are receptor proteins because they can specifically bind certain target molecules. So here we have the green molecules that are being bound, and when they actually bind, this actually activates the membrane. It provides a signal for the membrane to start invaginating. And then, of course, formation of the vesicle will internalise the receptors with their bound molecules. All right, so you can see here that this process of endocytosis means that little pieces of the plasma membrane are being internalised. So that means that the plasma membrane is getting smaller and smaller as the process occurs. So this is balanced by the opposite process, which is called exocytosis, meaning that vesicles are formed within the cell. Those vesicles fuse with the plasma membrane and then release the contents to the outside of the cell. So here we have a vesicle that's been formed inside the cell. So the membrane surrounding this vesicle may have originated from the Golgi apparatus, for example. It approaches the plasma membrane, and then the membrane of the vesicle will fuse. The plasma membrane opens up after fusion and releases this product to the outside of the cell. And once again, we can see this fantastic photo here taken with an electron microscope. illustrating that very process. And that's it for movement across membranes. How's everyone going?
Yeah, good.
Good? Terrific. I think because you've all done the prac, you're all very familiar with those concepts. Oh, we've got a few people on here. Oh, we've got Justin, Brad. Hello, everyone. All right, lecture 13, cells and tissues. And before we launch into this lecture, let's just have a 5-minute break, shall we?
Sounds good. Yep.
Okay, I'll see you all in 5. Oh, hello, Liz.
Hello from Singapore.
Oh, hello from Singapore. Well, some people are just lucky, aren't they? Oh, fantastic. I wish I was in Singapore. How long are you there for, Liz?
Just 2 days.
Just
I don't
2
know if you can
days?
see out the window. It's not exactly exciting weather. It's just hot and humid.
Oh, have you got your webcam on? No, I can't see. It's too bright outside.
Ah, okay.
Oh, lovely. I need to go on a holiday. Well, I did go to the US last year in April. That was lovely. I'm sure the living systems students didn't appreciate me taking off in the middle of semester, but it was okay. All right, let's make a start on lecture 13, cells and tissues. So we're going to take just a very general look at the major tissue types in animals and then in plants. So just a general overview. And I'll just get rid of this drawing, which is annoying. So let's start by considering a very young embryo, a very young animal embryo, or even a plant embryo for that matter. The embryo consists of a mass of cells, and these cells are said to be undifferentiated. That is, they haven't taken on a particular form or particular function. But very early on in the development of the embryo, certain cells will start to play specific roles, take on specific functions. And of course, all those cells have exactly the same DNA. They have the same genetic code in the form of their DNA, but though the genes contained within the DNA are expressed differently in different cells. So cells are said to differentiate during development. And specific types of cells might aggregate to form tissues. Tissues might combine to form organs, and then in complex animals and of course in plants, organs might combine to form systems, that is, systems that carry out entire processes. So in plants, of course, we can think of the respiratory system, the digestive system, whereas in, sorry, did I say in plants? I meant in animals. In animals, we can think of the respiratory system or the digestive system. In plants, we can consider the vascular system or the reproductive system. Okay, in animal cells, the plasma membranes of adjacent cells may be separated from each other. They might not be in close contact, and where they are separated from each other, physically separated, in between those cells, you'll find what's called an extracellular matrix. The extracellular matrix is a fluid network of proteins found within a highly hydrated polysaccharide gel. The extracellular matrix is secreted by specific types of cells. So let's consider the proteins, the major proteins found in the extracellular matrix, which I'm going to abbreviate to ECM. The proteins fall into two broad categories: structural proteins and adhesive proteins. Structural proteins include collagen and elastin. Collagen is probably the most abundant protein found in animals. It can constitute up to about 25% of the total protein in animal systems. Collagen is a very tough protein. It has very high tensile strength, meaning that it can be stretched to a certain degree without breaking. And in fact, the tensile strength of collagen has been compared gram for gram with the tensile strength of steel. So that illustrates how tough collagen is. So collagen has high tensile strength, but it has very low stretchability, which is referred to as elasticity. Now, in contrast to the collagen protein, elastin proteins have very low tensile strength but high elasticity. So to picture this, we can take the skin on the back of our hands and just pull that skin up from the back of your hand. So I want everyone to do that. Pull up the skin on the back of your hand and tell me which one of these proteins allows you to actually stretch your skin.
Elastin.
The elastin, yep. So it's the elastin that enables you to stretch the skin, but which protein prevents you from tearing the skin off the back of your hand? The collagen, yep. The collagen is the one with the high tensile strength. Okay, let's look at adhesive proteins. Fibronectin, labenin are just two examples of adhesive proteins, and these are proteins that mediate the attachment of cells mostly to the extracellular matrix itself, but also mediates attachment of cells to other cells. All right, the most abundant polysaccharide that's found in the ECM is the glycosaminoglycans. Glycosaminoglycan polysaccharides contain a strong overall negative charge, and this functions to attract cations into the extracellular matrix. As the concentration of cations builds up, then water follows simply by osmosis, and this is how we end up with this highly hydrated extracellular matrix. Proteins in the extracellular, in the ECM, integrate cell activities and guide cell movement. This is more relevant to animal embryos, where during the early embryonic development, certain cells will actually move, detach and move through the embryo to a different part of the embryo, and that movement is actually facilitated by the extracellular matrix itself. There are two major types of ECM. One is called the interstitial matrix, which is found in connective tissues, and one is called the basal lamina, which is found under epithelial cell layers. And we'll come back to some of those terms in just a minute. All right, let's look at the two major types of ECM, which we can see here. So at the top here, we've got a single layer of cells, which are called epithelial cells. And then just below that, we've got this brown region here, which represents one type of ECM, the basal lamina. And then below that, this region here is called the second type of ECM, which is called the interstitial matrix. And we can see within this particular ECM, there are very few cells. So we've got one type of cell here, which is referred to as a fibroblast cell. These are cells that are specialised for secreting the ECM itself. We've got a couple of cell types. These are cells of the immune system, a mast cell here and a macrophage. And we have one blood capillary here. But mostly we just have ECM, and we can see different types of fibres. So we've got collagen fibres here, elastin fibres here, and these fibres down here are the glycosaminoglycans. So that puts it into perspective here, and we can see that illustrated also to the left. BL is the basal lamina, E, epithelial cells overlying the basal lamina, and at the bottom here we have the interstitial matrix of connective tissue. All right, where we have cells that are very closely connected to each other, then the plasma membranes of those adjacent cells are connected by molecular complexes called junctions. There are three main categories of junctions: tight or occluding junctions, which prevent passage of molecules in between the cells. The second main group are the anchoring junctions, which provide sites of attachment. And then we have communicating or gap junctions, which is specialised for communication between cells. All right, that's illustrated here. So here we have one cell, one epithelial cell of the intestine. And it's obviously been cut away so that we can see the inside of the cell. Here's an adjacent cell, or part of the adjacent cell, and part of an adjacent cell on this side also. So let's look at the junctions. Here we have an example of a tight junction right there. Here we have one type of anchoring junction called an adherens junction, another type of anchoring junction called a desmosome, a communicating or gap junction, and another type of anchoring junction called a hemidesmosome. So you can see that this one down the bottom is anchoring the cell to the underlying basal lamina. So one type of extracellular matrix can be seen down here. But if we look at the inside of the cell, we can see that the junctions are mostly associated with some filaments. So in green, we can see filaments running along the inside of the cell, and these will attach also to this anchoring junction on the other side. We can see brown filaments here that are connecting this anchoring junction with other junctions. Here, there. So there seems to be connections between most of these junctions. Does anyone remember when we're talking about these filaments inside the cell, what do they actually make, what do they make up inside the cell, these different types of filaments? So I'll give you an example. Some of the filaments are called intermediate filaments, others are called microfilaments, and another type is referred to as microtubules, and they all collectively make up what?
The cell skeleton, was it?
Yep, correct. The cytoskeleton. Yep, those different types of filaments that we're looking at make up what's called the cytoskeleton within the cell. All right, so let's start with the first group of junctions, the tight or occluding junctions. These function to prevent the free movement of molecules through the fluid in between the cells. So once again, we're looking at an epithelial cell of the intestine. And this region here is the inside of the intestine, referred to as the lumen. So of course, This is where large food molecules are broken down into small molecules. Here we have a molecule of glucose, which is prevented from moving in between the cells. And it's prevented from moving in that direction by the presence of this tight junction. All right, instead...
Would you be able to change the size of your PowerPoint down to 75 or something so that we can see the bottom of this PowerPoint?
Yeah, sure. Yep, let me try that. Is that better?
Slightly.
Slightly. So you still can't see the bottom?
Possibly not.
Okay, can you see where my pointer is?
No.
No. Okay, all right. Let me try going a little bit.
Just
go
70. Type 70 and see where we go.
I'll go 70. No, it won't even let me go 70. Oh, that's interesting.
I can see the bottom of it, but I've got it on full screen view.
Yeah, I can see it
right.
too.
Oh, so you can all see the bottom?
Yeah, I can.
Okay. Andrew, have you got full screen view? Or have you got it minimised maybe a touch?
I believe I'm on full screen, yeah.
Oh, that's interesting. Can everyone else see the bottom of the slide?
I can see most of it now. Just even changing it to 75 did make a big difference because I couldn't read any of the box that was there before. So
oh, right. Okay. Can you see my cursor now, my hand?
No.
You can't?
No.
Ah, okay.
I can see the glucose carriers. That's the
So you can see
that.
Just that, but nothing further down.
Yeah.
Okay, that's interesting, but everyone else can see it.
Yeah, mine's all right. It could be that Andrew's monitor's a bit smaller or his resolution's a bit lower.
Right. Yeah, I'm not sure. I haven't had that problem before.
That's better anyway. Thanks.
It is better? Okay. All right. Sorry, Andrew, that I can't help you with that. Okay, so let's look at our glucose molecule. In order for glucose to actually move into the cell, it has to be actively transported by membrane transporters that are found in the plasma membrane here. They're not shown here, but they do exist. They're active glucose transport proteins. So they move glucose into the cell, and these active glucose transporters are found only on this side of the epithelial cell. So active transport of glucose into the cell against the glucose concentration gradient. And then on this side of the epithelial cell, there are passive glucose membrane transporters. So then glucose simply moves passively down its concentration gradient from high concentration inside the cell to low concentration in the bloodstream. So the tight junctions function also to restrict the membrane proteins to a specific area of the plasma membrane. Okay, our second major group of junctions, the anchoring junctions, and there's quite a few different types of these. They function to provide mechanical support, mostly to connect cell to cell or cell to extracellular matrix. Here are some different types of desmosomes, which are connected to intermediate filaments of the cytoskeleton within the cell. Hemidesmosomes, which function to anchor cells to the extracellular matrix, and various types of adherens junctions. And there's a couple of different types here. Adhesion belts, which connect to actin filaments, so another type of filament of the cytoskeleton, and focal junctions. And you don't have to memorize all these different types of anchoring junctions. All right, but they are illustrated here for you. So once again, Here's a type of anchoring junction, adherence junctions, desmosome, and hemidesmosome at the bottom here. All right, and our third group of junctions are the communicating or gap junctions. These are specialised for electrical and chemical communication between cells. They provide a pathway of low electrical resistance and permit the rapid current spread between cells. So here we have communicating junctions illustrated here in green. So this is represents the plasma membrane of one cell, and down the bottom here we have the plasma membrane of an adjacent cell. So you can see here how these communicating junctions will link one cell to the other. All right, now let's take a look at the major tissue types in animals. There are four major categories of tissues. The first one shown here is the epithelial tissue. So epithelial tissue forms continuous layers as surfaces. So not just continuous layers around the body of the animal, but also continuous layers lining internal cavities or internal tubes. So for example, lining the digestive system. Epithelial cells that make up these tissues are bound together by tight junctions, and we saw an example, and also anchoring junctions. They function primarily in protection, but also in regulating the exchange of materials and secretion, depending on the type of epithelial cell. Epithelia are categorized in two ways. One, by the number of cell layers. If there's only one cell layer present, then it's referred to as simple epithelia. If there's more than one cell layer present, it's referred to as stratified epithelia. Epithelia are also categorized by the shape of the cells. So flattened epithelial cells are called squamous epithelia, cube-shaped, cuboidal, column-shaped, columnar. And we'll see some examples of those on the next slide. All right, so in this top panel here, we have simple epithelium. So remember, just one layer of cells. Squamous simple epithelial cells are these flattened cells. So because they're very flat, this provides a thin layer across which diffusion can readily occur. Cuboidal simple epithelial cells, as you can see here, again, just a single layer, cube-shaped cells, and these tend to function in absorption and secretion of substances. Columnar epithelial also function in secretion and absorption. All right, our next group here, the stratified epithelial cells. So stratified, remember, multiple cell layers. Here we have squamous stratified epithelial cells. Some of you might have seen two or more of these cells when you prepared cells from your the inside of your cheek. They were examples of squamous stratified epithelial cells. And here we have pseudo-stratified columnar cells. And these are quite unique in that some of these cells are quite specialised in secretion of mucus. So these might be cells that are lining parts of the respiratory tract. And these cells here, these specialised epithelial cells, secrete mucus, and the function of that mucus is to trap any foreign particles, dust, bacteria, viruses, and so on. So it's basically the first line of defence. Then other cells that we find here, epithelial cells here, develop all these little projections, like little hairs, but they're not hairs, they're projections of the plasma membrane, and they are called cilia. And these little cilia wave back and forth, and they function to move that mucus with its trapped debris, move that mucus up to the back of the throat where it can be swallowed. All right, our next major tissue type is connective tissue. Connective tissue tends to have lots of extracellular matrix. Less cells, more ECM. Connective tissue functions mostly to provide structural, metabolic and defensive support. Connective tissue is very diverse in its structure. It could be calcified, as you would find in bone and teeth. Connective tissue could be fluid, as in blood. All right, so it's just some examples listed here of connective tissue: bone, cartilage, blood, adipose or fatty tissue, and fibroblasts. So as we just said, components of connective tissue, the extracellular materials, or the ECM, is usually more abundant than the cells themselves. Our next major tissue type, muscle tissue. And of course, muscle cells are capable of contraction. And this is because they're composed of actin proteins and myosin protein filaments. They function mostly in the movement of the animal and in the movement of internal organs. Two major types of muscle tissue: striated muscle, which is highly organized, and smooth muscle, which is less regularly arranged than the striated. And some examples here of striated muscle includes skeletal muscle and cardiac muscle. And our last major tissue type in animals is nervous tissue. Nervous tissue consists of nerve cells, which we call neurons, and of course these carry information throughout the body of the animal. Let's look at the structure of a neuron. And here's one neuron shown down here. So we've got the main part of the cell here called the cell body, and we can see the nucleus here. Then we have these long projections from the cell. This is where the information is received by the neuron, and these are called dendrites. Then we have this very elongated part of the cell, which is called the axon. Now, as you know, neurons are very long-lived cells. They're mostly unable to divide to form new cells. So once they're formed, they tend to remain functional for the lifespan of the organism. Even though there is some evidence that's come out over many years now that have shown that there are some pockets of neurons that can divide, can replace damaged neurons. But mostly once they're damaged, that tends to be it. So because they're long-lived cells, they are supported by a different type of cell called glial cells, which you can see here wrapped around the actual axon of the neuron. And these glial cells also give rise to what's called the myelin sheath, which maintains the composition of the extracellular environment. All right, and here we have the four major tissue types: nervous tissue, epithelial, connective, and muscular tissue, and the location of some of the different cell types within these tissues. All right, let's move on now from animals to plants. And of course, plants are quite different in that plant cells have rigid cell walls to the outside of the plasma membrane. And this limits the size and the shape of cells. And unlike animal cells, plant cells tend always to be found very close to each other. Even though there are some gaps, some fluid regions in between some cells, we don't have that extracellular matrix as you find in animal tissues. All right, so rigid cell walls allow plant cells to be surrounded by a hypotonic solution, which we saw in the previous lecture. So without that cell wall, then the inflow of water by osmosis would cause plant cells to burst. Cell walls are also important in plant metabolism. They contain lots of different enzymes. Cell walls are important pathways for transport of water, transport of dissolved mineral nutrients. They're important in absorption and secretion of many different substances. And of course, they act as an important barrier against pathogens. Let's look at the structure of a plant cell wall. Cell walls are composed primarily of cellulose, our structural polysaccharide called cellulose. And you might remember from one of our earlier lectures that cellulose is a polymer consisting of many glucose molecules linked together into chains, and these chains are organised into microfibrils. Okay, don't worry too much about the rest of this information here. We'll go into more detail about this in second semester in organic chemistry. Cellulose microfibrils are embedded in and linked to a matrix of other polysaccharides and also substance called pectin and various proteins. So let's look at that down the bottom of the slide here. So in green, we have our cellulose microfibrils, and these are connected to various proteins, which we see here in purple. Then we have in red, pectin molecules and various other polysaccharides. So these shorter green rods here. Okay, what about cell walls of other organisms? Cell walls of fungi are primarily composed of chitin. So this is one of the main differences between plant cells and fungal cells. Cell walls of bacteria are composed of a different substance. And this, the molecule that's mostly found in bacterial cell walls consists of two modified polysaccharides, and these are linked by amino acids. Once again, this information is here just for your interest. All right, so very briefly on how cell walls develop. So when one plant cell starts to divide to form two new cells, that is, it's undergoing cell division, then new plasma membrane will develop between the two new cells, and then the cell wall material is added in between those two newly formed plasma membranes. Then in between that new cell wall, in between the two new cell walls of the two new cells, a region called the middle lamella develops, and this is where you'll find lots of those molecules called pectin. Most plant cells will develop an initial thin cell wall called a primary cell wall. And then some plant cells also develop a second layer called a secondary cell wall. And the secondary cell wall is strengthened by the presence of this substance called lignin. Okay, so also concepts that we'll come back to in other lectures and in other subjects. Okay, so because plant cells have rigid cell walls, this limits contact between the adjacent cells. But plant cells are in contact with each other, well, most of them are anyway, and that contact is facilitated by the presence of plasmodesmata. So let's look at them here. So you can see these little channels here connecting the cells These are called plasmodesmata. Some plant cells have lots of plasmodesmata. Others have plasmodesmata restricted to one side of the cell or to the other. So it really just depends on the cell type and what its function is. Plasmodesmata are channels that link plasma membranes and the cytosol of adjacent cells. So these channels, these plasmodesmata, are bounded by the plasma membrane contributed by both cells. Within the plasmodesmata runs a narrow tube called a desmotubule, and the desmotubule is actually continuous with the endoplasmic reticulum. So the desmotubule is connecting the endoplasmic reticulum of one plant cell with the ER of the adjacent plant cell. So there are actually lots of connections between plant cells. All right, let's look at the major tissue types in plants, of which there are 3. But before we do, let's look at where these tissues are actually derived from. So in plants, there are 2 major regions of cells that are undifferentiated, and these cells are constantly dividing. And the division of these, the constant division of these cells enables plants to grow. So these regions of undifferentiated cells, which you can consider as being analogous to stem cells in animals, are called apical meristems. The apical meristem is a specialised region at the growing tip of a root or the growing tip of the stem. They're continually dividing undifferentiated cells, and we often refer to them as meristematic cells. Just think of stem cells. All right, so at the very tip of the growing shoot, we have what's called the shoot apical meristem, abbreviated to SAM. At the growing tip of the root, or rather just behind the growing tip, we have the root apical meristem, and this is abbreviated to RAM. Here we have a scanning electron microscope image of the shoot apical meristem. So it's found just here. It's just a small group of cells here, undifferentiated cells here, and each of those cells will divide to produce 2 cells, and those 2 cells are called daughter cells. One of the 2 daughter cells remains undifferentiated, so it remains as part of the meristem, and the second of the 2 daughter cells will differentiate to form part of the mature body of the plant. So, for example, cells that differentiate from the apical meristems might give rise to dermal tissue. So that's the first major type of plant tissue. Other cells that differentiate might give rise to ground tissue, and other cells might give rise to vascular tissue. All right, so let's start with dermal tissue. Dermal tissue forms the outer covering of a plant. So this would be similar to epithelial tissue in animals. But rather than calling the cells epithelial cells, in plants we call them epidermis or epidermal cells. Epidermis is the major type of dermal tissue. It consists of one or several layers of closely packed cells, and epidermal cells are quite specialised in that they secrete a layer, a waterproof layer of a substance called cutin, which is a lipid substance. And this layer that forms over the entire surface of the plant is called a cuticle. And obviously, being a waterproof substance, it prevents water loss from the plant. The epidermis also contains tiny pores that are called stomata. And it's because of these pores that the important carbon dioxide can diffuse into the actual air spaces within the leaf, and that carbon dioxide is essential for the process of photosynthesis. So stomata function to allow the exchange of gases. And I don't have an image here, but we will talk more about stomata, I believe, in second semester from memory, or maybe even still this semester. All right, the stomatal pores are surrounded by two specialised cells called guard cells, and these guard cells actually control whether the stomatal pores are open or closed, and that depends very much on environmental factors. All right, so let's look at leaf, and just let's look at that tiny little boxed section there, enlarged here. So we can see, well, we can't actually see it, but there's a layer over the surface of the leaf, which is called a cuticle. So remember, this is not a layer of cells, it's just a layer of cutin, and that cuticle is secreted by these epidermal cells here. And below the epidermal cells, we can see another layer of cells, which is not part of the dermal tissue, and these are photosynthetic cells. All right, epidermal cells. Here's one type of epidermal cells, and these are specialised in that they contain what we normally refer to as leaf hairs. They're not actually hairs, they're extensions of epidermal cells. And here we have some oil glands. Sorry, these ones are the ones that are extensions of epidermal cells. These particular leaf hairs are referred to as trichomes. All right, second major type of plant tissue is called ground tissue. And there are many different types of ground tissue cells, which we won't go into here. This is just a very broad overview. Ground tissue includes storage tissue and structural tissue. Here are some of the functions of ground tissue. Photosynthesis, so those photosynthetic cells that we just looked at are examples of ground tissue. Ground tissue functions in storage, aeration, support, and strength. And finally, the all-important vascular tissue. So if we think about vascular tissue in animals, we would consider the blood circulatory system and the lymphatic system. In plants, the vascular tissue functions to transport water and dissolved mineral nutrients, and also carbohydrates that are synthesized as a result of the process of photosynthesis. Vascular tissue includes two different tissue types: xylem tissue, which functions to transport water and minerals from the soil solution into the roots, and then always in an upwards direction through the plant to the leaves. Second major type of vascular tissue is phloem tissue. And the phloem transports the products of photosynthesis, which of course are carbohydrates, and we refer to them as photosynthates. These are transported from where they're synthesized, primarily in leaves, to places of use or storage. So while xylem transports water and minerals always in an upwards direction, phloem will transport photosynthates in both an upwards and downwards direction. Okay, here we have a section of a stem from a plant, and if we cut through in this direction, cut a section in this direction, which is called a cross section or transverse section, and then just flip that over so we can actually see the cells within the stem. And you can see these little bundles of cells that have been coloured, and they're referred to as vascular bundles. So these vascular bundles contain both cells of the xylem tissue and cells of the phloem tissue, and they're always associated with each other within these little bundles of cells. If we enlarge one of those vascular bundles, we can see here cells of the phloem in blue and also cells of the xylem in red. And you'll learn, those of you that go on to do plant physiology will learn a lot more about these important tissues and how they function. All right, and that concludes our lectures for tonight. So let's look at your next task. Tutorial number 9. Tutorial questions on both of these lectures are assessed, and they are due by next Monday at 6 p.m. As normal. All right, so does anyone have any questions for me? And I have a question for you all. Has everyone completed the online test?
Yes.
Yes.
Yes? Terrific. So remember, if you haven't, it closes at 11 p.m. tonight.
Mm-hmm.
Which means that you probably should start that as soon as we finish the class tonight. Okay, because I did notice a couple of hours ago that there was still a few students that hadn't completed the test. All right, so really important that you get that done.
Mm-hmm.
Okay, so any more questions for me?
No.
No? Everyone good?
Yeah.
All right, prac reports. Has anyone made a start on their prac reports? So prac workshop students, remember you need to write 2 prac reports. Please refer to the instructions given in the last part of your prac manual. And if you have any questions, let me know. Any questions at all? Feel free to send me a draft of your first report if you would like some feedback.
Okay.
But of course, that draft has to be emailed to me at least 1 week before it's due. Otherwise, I can't guarantee that I can give you feedback on it.
Mm-hmm.
Okay. Also remember that you need to write, one of those reports must be written on the acid-base titration prac, and the other report can be written on any prac of your choice. Okay. All right, so we're all happy?
Yep.
I don't know if that's the right word, but Liz, enjoy Singapore.
Thank you very much. Back down under next week.
Okay. Terrific. All right, thank you, everyone.
Okay, thank you.
Thanks, Savannah.
Thank you. Thank you. I'll talk to you all next week.
Bye.
All right. Good night.